I thought I'll make one comment before I make a brief presentation to you about AFib. Then we'll go back to Natalie's case to close up, and at noon, we'll actually have the lunch and the panel discussion. The slide that I put up over here I thought would be of interest for the audience here, mainly because this actually deals with what we are now beginning to see in more and more cases where patients come to us after failed AF ablations. And what you see over here is percutaneous epiendo mapping during AF ablation. So we actually do epicardial mapping. And if you look at this case, you can see that the endocardium is completely, scarred, whereas epicardial mapping in the same case actually shows that you have a lot of viable myocardium. And this is, of course, epi mapping post creating a box lesion endocardially. You can see that the epicardium is now no longer activated. So this is a kind of a very specialized kind of a procedure. It is still useful in patients who have redo AFibs. It is investigational, and our group and others are in the process of doing that. And I think this is also the basis in which we are now, for the first time in the EP lab, instead of surgery, in the EP lab, we are trying to test the mechanism of AFib, especially the sandwich hypothesis, which Moritz Alessi and his group have shown, to show epi-endo differences in activation in atrial fibrillation. So this whole concept is being looked at in the EP lab. And one other point that's worth keeping in mind is, since esophageal A fistula formation is a huge risk in AFib, especially the posterior wall, where you can actually get the fistula. One of the other approaches that we have used, in addition to what you just saw Dr. Natali showing you moving the esophagus, you can actually stick the pericardium and put in a balloon in the pericardial space. So this is LAO, and if you look at it in the RAO view, so this actually creates a physical separation of the esophagus and the atrium, and then you can also...
Y en ese caso podemos también hacer ablación.
All these measures are to protect the esophagus. So let's spend a few minutes now, because I was asked to address autonomic nervous system and atrial fibrillation. By the way, the slide is supposed to show...
La diapositiva debería...
it's kind of spread out. So let me make the initial comment. All the people you see here, part of my team, in fact, one of my colleagues, Dr.
Uno de mis colegas,
here, and our lab manager from Colombia, Mr. Eric Asensio, is here. These are the people who do all the work, and I get to give the talks. So we thank all the groups that have actually funded this research, and most of this, of course, comes from research grants from the National Institutes of Health in the US. So we love interventions. We live in two worlds. We do complex interventions, and we're trying to do a lot of basic science to really understand how nerves control the heart. The UCLA has a group of investigators shown over here, and our team's effort has been really to look from molecule to man to understand how nerves control the heart, which is why the Neurocardiology Research Center of Excellence is a part of the Cardiac Arrhythmia Center. In fact, it's a research arm. We also have a collaboration with these investigators from Caltech, Nebraska, UCI, Oxford University, Oregon Health Sciences, and Harvard. And you'll be seeing data from some of these groups. And this is the consortium that we have, which is a comprehensive cardiac mapping consortium. So how is this all relevant in the world of AFib? So the entire field of AFib, 20 years ago when we were doing this procedure, we used to sit and map for single PACs. And for some of the trainees and the fellows and the younger colleagues in the audience, we are now dating ourselves. And this is the kind of cases we used to do. We would sit down for hours, look for PACs that initiate AFib. And of course, there's been a lot of, what was started off as focal AFib, the strategies for ablation have evolved over the years. All the way from, we used to do wrong things in the early days by trying to ablate foci within the veins. And slowly the whole field has now moved into a form of electrical isolation of essentially the posterior wall of the atrium and the pulmonary veins. So, and the technology, the catheters, the whole procedure has evolved quite a bit over the years. So where are we in 2017? What are all the strategies available for AFib? You could do a surgical maze. You can do some form of pulmonary vein ostial antral isolation, many variants of left atrial catheter ablation, mitral lines, linear lines have been added to the procedure. There's ablation of complex potentials. For a period of time there, rotor ablation came and it went. And of course, what has always been talked about is, are we causing some kind of an autonomic modulation because of AFib? And I'll try to address that. So this again is another image from the McAlpine collection, which Dr. Sands had already pointed out. And this is a, this is a beautiful image of the right atrium, the left atrium, and the complexity of the anatomy of this area. What are we doing with AFib? We are doing a variety of diverse procedures. Interestingly, it still works in a subset of patients, but how and why it works is still a bit of a mystery. We saw ligament of Marshall ablation, which is, interestingly, many of these interventions may also be modulating the autonomic nervous system. So a comment about mechanism of AFib. These are some of the truths which I think most people in the world would agree to. Mechanism of AF in humans is poorly understood. Mechanisms are very poorly understood in humans. The benefit of ablative approaches is not clear at all. So that is another problem. And of course, use of ablation is increased. The challenge we're dealing with AFib, as you saw even in the cases, live cases we are showing you today, post-mitral valve or aortic valve, post-valve surgery, complex substrates. So how do you deal with this? You saw one case, which is a cryoablation, which is paroxysmal fib, structurally normal heart, completely normal anatomy. So the patient is having their first procedure. So you see this entire spectrum of patients. So this is the challenge of the field. Of course, if we can understand a mechanism, then we could actually go target the procedure beautifully, right? If you really understand, if you have a focus, or let's say it's a functional problem, we are not even able to classify atrial fibrillation from a scientific perspective to help us choose therapies. And of course, if we had that, we can put this into this complex pathophysiology of what happens in AFib. Now, there have been many landmarks in fibrillation research. This starts off over 100 years ago, when minds actually came up with anatomic reentry. They were working with rayfish atrial rings and turtle hearts. Walter Gary, working at Barnes Hospital, Washington University, St. Louis, proposed the critical mass hypothesis of ventricular fibrillation. Gordon Moore created the multiple wavelet hypothesis based on actually a mathematical model. Moritz Alassi showed that functional reentry could be the basis of arrhythmias. This is, again, now almost 45 years ago. Then came the mathematicians. The mathematicians came up with this whole spiral wave theory, which, of course, many experimentalists immediately caught on to. And Dr. Halife's group had done optical mapping, and they were able to show in a small subset of patients when the heart was mechanically uncoupled, they were able to actually demonstrate rotors in their system. And of course, multiple wavelet fibrillation is what has been consistently seen both in electrical and optical mapping studies, again, for the past 40 plus years. There have been many other theories that have come up, which is the mother rotor fibrillation theory, which is what Pepe Halife's group talks about. And our own colleagues at UCLA, Dr. Weiss and Zealand Q, have shown that VF and AF may be mixed focal and reentrant, which is due to delayed after depolarizations or early after depolarizations. So where do we stand? We are like the blind man and the elephant. So we are stuck with having many theories out there. And in fact, someone once joked about this. They said, dogs have fleas and professors have theories. So these are all the theories we are. So it doesn't help us as clinicians. So we are still left with having to really deal with these two worlds which are still not connecting somewhere in the middle. And the whole conundrum has been sort of expanded, if you will, by having more and more mathematical modeling studies without adequate experimental data for us to interpret what happens in its simplest format, which is anatomic reentry, which we actually understand fairly well, and we're able to map and ablate. Everything else in this sequence is a bit of a conjecture and not having good strategies of how to ablate. The reason why ablation has become so challenging for us in this field is also because of the fact that when you map fibrillation, interpretation of mapping is not easy. It's very complicated. Just look at this example. I briefly alluded to this slide earlier today, but look at how mapping looks, and you actually need a lot of electrodes, and you need a lot of computer processing power to make these interpretations. Whether it's a single source, which can be automatic, mother wave, fixed rotor, moving rotor, or multiple sources where you can have multiple foci, unstable circuits, multiple wavelets, or sometimes you can have a focus with multiple wavelets, which occurs in the periphery of regions in terms of conduction. So we live in a very complicated world in terms of what you see in mapping of fibrillation. So where does the autonomic nervous system fit into this? This, we know, has a very important role to play because it's based on clinical experience. We know it plays a role, and it's being used for managing fibrillation, in some cases with reasonable success, as you'll see. To understand the autonomic nervous system, and I'll show this slide again once tomorrow, the heart is like any other organ in the body in terms of how it's innervated. It has neurons sitting in the heart, which is the little brain of the heart, which sit in the fat pads, right? We're going to talk about that. The heart also connects to the extracardiac stellate ganglia. So this forms a heart stellate ganglion heart loop. The heart also sends impulses to the spinal cord. Process there comes back to the heart. So you get heart, spinal cord, heart, and last but not the least, heart, brainstem, heart, which is the vagal nerve, the vagus complex. And now, as it turns out that the spinal circuit also can be modulated for managing atrial fibrillation, and spinal cord stimulation is one of the therapies. So this is not just of theoretical interest, it has practical applications. The nervous system itself is pretty complex. I'm not going to waste time telling you about this slide, but I'd just like to highlight the fact that the heart itself has fat pads, which actually have a lot of neurons that sit in them. And these neurons together form what is called the little brain on the heart. So when you see in the literature, when you see papers that say fat pads, they're dealing with these neurons. When you hear about the stellate ganglia, which itself is a very complex structure, and we use that, of course, we cut that to manage ventricular tachycardia, that, when we're talking about that, we're talking about this group of cells. So what about anatomy? You have to go back for several hundred years to look at anatomy images of the heart. And these nerves that come to the heart are a subject of intense investigation now in 2017. When you look at all these anatomic images in any of these books and publications and slides that you see, there is a big question that has come up, because some of these fibers which are marked as nerve fibers on textbooks may not be nerves. Some of them may be lymphatics. These have not been studies where people have done the kind of careful studies histologically to show that this fiber, if it's being traced to the heart, is indeed a neural fiber that is going to the heart and not some other kind of a vascular or a lymphatic structure. But what we do understand well is at the stellate complex and at the level of the heart. What is between the two is where there is a lot of controversy. But what is seen at the heart? The heart itself has neurons that sit on the fat pad. These are very important because you see a lot of papers where people are trying to ablate these fat pads. And these fat pads is where the little brain on the heart sits. And these can be modulated. We can record from these neurons. And the understanding of these neurons is improving day by day. Where do they sit? They sit in designated areas, like collections of neurons within these fat pads, and each one of them control a distinct anatomic area. The right atrial ganglionated plexus, for instance, controls the SA node. The RVGP, the ventricular GPs, which are just around the base of the heart, near the coronary arteries, control the ventricles. And you'll also see that the posterior atrial wall, where we are doing a lot of ablation, also has a lot of ganglionated plexi, especially the dorsal atrial ganglionated plexus. All this is very interesting for us as electrophysiologists, because when you see a complex system, you don't have to get disheartened. It actually tells you that you have many angles of attack. And what you see over here is various approaches that have been used to modulate arrhythmias. The vagal nerve stimulation, tragal stimulation, just stimulating the ear because that connects to the vagal complex, and of course, all along the spinal cord, and also renal modulation, as we'll see. So you can do a whole lot of procedures, and you just saw today in the live case, which is ablation of the vein of Marshall, which, by the way, has a lot of autonomic structures. So ablating the vein of Marshall is also a form of GP ablation. So let's start off with the most extreme or a bookend example. Many years ago, my colleague Marmar Visegi actually did this study. This is published in JACC, I think this was in 2006. We were impressed by the fact that you never see atrial fibrillation in a transplanted heart. So the only time you see AF in a transplanted heart is when the heart is inflamed and there's ongoing rejection. So it's extremely uncommon. In fact, you may see macro-reentrant tachycardias, which need to be mapped and ablated, but we never ever saw AFib. And in fact, when we looked at this data more carefully, AFib in stable patients was almost unheard of. And of course, when you do see what looks like irregular atrial tachycardia, it was more often than not just inflammation of the heart. And what you needed to do was to give them immunosuppressives to control the inflammation. A structure that has been well studied in the experimental lab and by Dr. Peng Chen, and this is a slide courtesy Dr. Chen and Dr. Fishbein I showed you earlier during the live case. This is the vein of Marshall, which we saw very beautifully ablated by Dr. Valdrabano and Garcia. This structure, when you look on the outside of it, on the epicardial aspect, it actually has a lot of GPs. So when you saw the points that was made by Dr. Garcia and Valdrabano, in your mind, when this has happened, you also have to remember that this ligament of Marshall actually has these autonomic ganglia that are being ablated and modulated by alcohol injection. Of course, we actually saw a component of electrical impulse propagation in the atrium also resulting from targeting the structure, but it also is a very rich autonomic source. This is the study that Dr. Valdrabano made a comment on, and I'm not sure if he's back from his case, but he's outside, or maybe he should come here and make a comment on this. This is a beautiful study, and the reason we like this is because it's a human data set. And Dr. Valdrabano's team has actually done this study where they've used microcatheters. Today, he had to just use a guide wire to get you a recording, but he's actually placed the cardema, the microcatheters within the ligament of Marshall, so you can actually get recordings from it. And in this case, they've actually done high-frequency stimulation to induce AF. And you can see that post-ethanol ablation, when you do the same high-frequency stimulation, you actually do not get fibrillation. So this actually shows that you modulated the entire structure so that the ability to induce AFib itself has come down. So this is it's one of the papers from his group on modulating the ligament of Marshall for controlling atrial fibrillation. And this is one example where they actually show a regionalized parasympathetic denervation of the human left atrium. So remember that ligament of Marshall intervention has a structural component, but it also has a very critical autonomic component. The other exciting data that came out in the field, in the literature, is this data set. This comes from a group in Russia where post-op, when they came off pump, they actually injected botulinum toxin, Botox, at all these fat pads, which I showed you just in the previous slide. And what was remarkable in their study was they found that compared to placebo, when you had Botox injection, not only did it have a big impact on post-op AFib, it seemed to have an effect that lasted up to a year. So this study has caused a lot of excitement, and there is a lot of ongoing research to try to understand why we have this benefit for injecting Botox. Does it alter neurotransmission within these ganglia? So that is an area of very exciting research that's going on. And we really need to understand this. And this is actually a smarter way of targeting the fat pads. You're not ablating it, you're not damaging it, but you're perhaps just modulating how the neurons talk to each other. So this is like giving a headache pill to deal with a headache instead of cutting off the head. So the other autonomic interventions that's worth talking about is, of course, renal denervation. The kidneys are very fascinating because if you stimulate renal nerves, you can record from the medulla. You can actually get electrical recordings. These afferents project to the higher centers, which control central sympathetic outflow. And there is some early data. Again, this comes from the group in Russia. Very preliminary studies where they've done renal denervation for AFib. And in a preliminary study, there are still ongoing studies where they showed that adding renal denervation to pulmonary vein isolation seemed to have a clinical benefit. Again, early studies, we have to see where this data falls in. What is happening in the experimental world? One experimental approach that is worth commenting about is, of course, spinal cord stimulation. And for some of you who are very nerdy in this audience, you should go read this book by Ronald Melzack, which is called The Puzzle of Pain. And this actually talks about the whole gate control hypothesis. This all comes from pain literature, and now all this is becoming very relevant to cardiac neuroscience. This is a slide that was given to me by Professor Foreman, who's one of our collaborators. He's University of Oklahoma. And Bob Foreman and his group have done a lot of work with spinal cord stimulation, where you actually have a electrical catheter in the dorsal spinal cord which stimulates the spinal cord, and this alters neural processing within the dorsal horn, and the net benefit is control of cardiac sympathetic output, and in fact I should say modulates cardiac output. Now this was actually tested in an animal model shown over here, where you do mediastinal nerve stimulation, which is the nerve fibers that go to the heart, and you can get atrial fibrillation. And in this particular example, this is a study published by Dr. Arma and Ardell in 2012, which actually shows that if you do spinal cord stimulation, you can actually prevent inducible atrial fibrillation. So this is AFib at baseline, this is AFib with spinal cord stimulation on. So this tells you that you can actually deal with an extracardiac structure and control atrial arrhythmias. And I think this is where the tracheal stimulation and the data that you're seeing in vagal modulation is going to be very interesting. And hopefully if some treatments like this come on board, there may be a convergence of effects. It's also thought that some of the data that you're seeing on exercise and obesity control and AFib may actually be working through an autonomic mechanism. So this is their data showing vagal nerve stimulation along the lines of SCS, showing that you can actually mitigate atrial fibrillation by vagal nerve stimulation. One additional advance that has been made in the field, and this is data again coming from Dr. Ardell's work, is that when you record directly from the neurons on a beating heart and compare preemptive vagal stimulation, you can see that the neurons and the way they fire within the fat pad, this is the same fat pad where Botox was injected, right? You can see that these neuronal recordings and the signature has completely changed, and you do not get atrial fibrillation. So there is a lot of convergence of opinion now that if you modulate nerves, you can control atrial fibrillation. And this, of course, is a more complex arrhythmia, and it's going to be interesting to see what data we get.